This video is a summary of the 4 different nutrient tests that you need to do as part of A-level biology in order to figure out which nutrients are present in a sample of food. In this video, we're only going through the qualitative aspects. There's a separate video in which I cover how to do quantitative analysis involving a colorimeter. The first test mentioned in the specification is the burette reagent test, and this is a test that's used to identify protein. But what it's actually testing for is the peptide bonds in protein. So it won't identify individual amino acids because these don't have peptide bonds in them. The first step is that you grind up your solid sample using a mortar and pestle and mix it with some distilled water. To this, you add some burette reagent, which is a mixture of sodium hydroxide and copper sulfate and potassium sodium tartrate. You shake all of these together, and the positive test would be that the blue solution turns a pale purple or violet. The next test that you need to know about is the test for reducing sugars. So this isn't a test for all carbohydrates because not all carbohydrates are sugars. The large polysaccharides like starch and glycogen are not. And then not all sugars are reducing sugars. So when we talk about reduction, this is a term from chemistry which means gaining electrons. So a reducing agent, like a reducing sugar, is something that causes other molecules to gain electrons and be reduced. And meanwhile, the sugar is itself oxidized, so it loses electrons. Now, in real terms, what this means is that our sugar needs to have an aldehyde group or a ketone group if we're talking about a very strong oxidizing agent. And the good news is that all monosaccharides have this group, so any monosaccharide will give a positive result for the reducing sugar test. So this is the form that glucose takes when it's put into a basic solution. So you can see that it no longer has that ring structure, it kind of turns into a long line, and at the end of that line we have the aldehyde group, which is what is going to be oxidized. Now, some disaccharides also have this group, so maltose does and lactose does, but crucially sucrose does not. So if you just do the basic reducing sugar test, you don't get a positive result for sucrose. To test for reducing sugars, we start out by grinding up the solid sample and mixing it with distilled water. We then add Benedict reagent. So this is made from sodium carbonate with sodium citrate and copper sulfate. So just like the burette reagent, it still has copper sulfate in it, but it now has an alternative base, and also we have sodium citrate. We shake the solution together and then we heat it to a very, very high temperature. Now, you're hopefully already aware from your GCSE biology studies that just saying warm it is insufficient because this test won't work without quite a high temperature. So realistically, your water bath is going to be at something like 70 degrees C in order for it to be hot enough for this to work. So the solution that you start with is a blue solution, and then our positive test is that depending on how much sugar is present in the sample, it will either become green, yellow, orange or red. And this is because it's forming a particular precipitate, and the more of it is, the further along the rainbow we get. I've made a separate video about this because colorimetry is a little bit complicated, but it's also worth you being aware that the Benedict test is the one that you can have quantitative data for. So one option is that you could filter your solution and weigh the precipitate that you achieve from that, although that obviously means that you need to make sure that there isn't any solid matter left in your solution to start with, because otherwise you would end up weighing that as well. And then the other thing you can do is you can take that solution and you can use it together with a colorimeter to determine the colour of the solution that's left, and therefore basically how much of the Benedict reagent you've used up. You can use the Benedict test to show that you have got the presence of a non-reducing sugar like sucrose or raffinose or stachyose. What you need to do is firstly do the Benedict test as we've just described and show that there is a negative result, and that rules out the fact that there are any reducing sugars present. Then you do some additional steps before repeating the test. And the reason for this is that we know that these disaccharides do contain monosaccharides. And so if we can hydrolyze the glycosidic bonds and break the disaccharide apart, we will then get back those reducing sugars. So when we then complete the Benedict test again, and this time we get a positive result, this tells us that initially we must have had a non-reducing sugar present. So the way that we hydrolyze these glycosidic bonds is by boiling the sample in dilute hydrochloric acid. We then add some sodium bicarbonate in order to neutralise the solution. And the reason for doing this is that those monosaccharides are only going to be in their reducing form with the aldehyde open rather than being part of a ring structure if they're in alkaline conditions. So having just added a load of acid in order to hydrolyse those glycosidic bonds, if we didn't do something to neutralise it, then the monosaccharides would not be in the correct form for the test to be administered. So having done those first two steps, we then just carry out the Benedict test exactly like we did before, and this time if we get a positive result, that tells us that we must have initially had a non-reducing sugar present in the sample. Pause the video and make sure that you can describe why it is that a student would heat a sample with hydrochloric acid if they were trying to carry out a non-reducing sugar test. Well, as we've just described, the function of the hydrochloric acid is to break the bonds in the carbohydrate. And since this is a carbohydrate, we know that these are going to be glycosidic bonds. So either our answer is going to be C or it'll be D. So the question then is, what are those bonds between? So we've got the options of glucose and sucrose or glucose and fructose. So we know that in chocolate there's going to be sucrose, and sucrose is made of glucose and fructose monomers. So therefore our correct answer is going to be D. The third test that you need to be able to describe is the iodine solution test. One thing that is worth noting is that at GCSE we probably just talked about adding iodine, but that's not actually what you're adding, because iodine is a kind of greyish black solid, and that's not what you add. What you add is iodine solution. So the most common way to make something that has iodide ions in is to dissolve some potassium iodide. So you just need to make sure you're talking about iodine solution or potassium iodide solution. In the absence of starch, this solution is sort of orangey brown, but in the presence of starch, it turns dark blue-black. Finally, we have the emulsion test for lipids. You might have also encountered other tests for lipids, like rubbing a piece of fatty food on some greaseproof paper, or perhaps the Sudan 3 test, but with the updated specification, you're now explicitly asked to learn about the emulsion test. So this is a test for lipids, and what we do is, to start with, we dissolve the sample in ethanol. So as with the other tests, if you have a solid food, you're probably going to crush it and grind it up in a pestle and mortar, and then add the ethanol together. You're going to shake those together so that the sample dissolves into the ethanol, and then you're going to pour it out of that tube, leaving behind any solid residue, and you're going to pour that solution into a second tube that contains water. The positive test here is that on the top of the water, a white emulsion forms, so kind of a white milky layer. Thank you very much for watching, and don't forget to check out the next video, which is a summary of how you can use a colorimeter to apply a more quantitative approach to these food tests. If you did find the video useful, then don't forget to like, subscribe, comment and share, and I'll be back soon with some more A-level biology.